DNA, or deoxyribonucleic acid, is a molecule that contains the genetic information an organism needs to survive. Each cell within an organism contains DNA. The structure of DNA is ladder-like and is twisted into a double helix shape. The sides are composed of phosphate and a 5-carbon sugar that are connected by covalent bonds. The rungs are composed of 4 nitrogen bases: adenine, thymine, cytosine, and guanine. The 2 nitrogen bases that make up one rung of a DNA strand are complementary to each other. The complementary base pairs are adenine and thymine, which are connected by 2 hydrogen bonds, and cytosine and guanine, which are connected by 3 hydrogen bonds. The order of these base pairs is what determines the instructions given out by DNA. The overall DNA structure is made up of a chain of nucleotides. One nucleotide consists of a phosphate, sugar, and one of 4 nitrogen bases. Replication is the process in which DNA duplicates prior to cell division within an organism. The process of DNA replication in all cells is called semi-conservative replication, where a new double-stranded DNA molecule is formed from the original template molecule. Helicase causes a separation or unzipping between nucleotides by breaking hydrogen bonds. DNA gyrase stabilizes the DNA double helix when helicase unzips the molecule. Then, single-strand binding proteins bind to single-stranded regions of DNA and keep the 2 strands separate so they can each act as a template for the new strands. The point where the splitting starts and the double-stranded DNA opens to make templates for the 2 daughter strands is called the replication fork. The leading and lagging strands are the 2 new strands produced during the semi-conservative replication process, and the 2 strands go in opposite directions with regards to their chemical structure. The leading strand is the first strand that is produced, which moves continuously in the 3' to 5' direction towards the replication fork. For the leading strand, first, a primer is produced from RNA primase at the replication fork. RNA primase is a type of RNA polymerase that synthesizes the primer. It creates a starting point for DNA synthesis. The primer, which is a short sequence of nucleic acids about 5 to 10 nucleotides long, lays a foundation for DNA synthesis, and it's complementary to a single-stranded piece of DNA, which serves as its template. The primer is the joining of RNA nucleotides that match the exposed DNA bases at the point of replication. The primer is only needed once at the very beginning as a hook for the DNA polymerase and is then removed. After there is a selection of nucleotides that have filled the section that has just been unzipped, DNA polymerase that comes in the variations 1 and 3, which are enzymes that are responsible for synthesizing DNA, add nucleotides one by one to the growing DNA chain, incorporating only those that are complementary to the template. This process is called continuous synthesis, where DNA polymerase 3 adds nucleotides to the 3' end of the leading strand. DNA polymerase 1 then replaces the primer with nucleotides. The lagging strand is more complicated. In DNA replication, DNA polymerase allows assembly only in a 5' to 3' direction, and the lagging strand runs in the opposite direction. In this process, RNA primase once again synthesizes for primers and lays down an occasional primer starting from the 3' end of the lagging strand. The DNA polymerase then comes in and works backwards along the strand, moving away from the replication fork, and sets down a short individual segment of nucleotides. Each individual segment is called an Okazaki fragment. This process repeats until the lagging strand is also complete. DNA polymerase 1 then comes in and replaces the primers. After this, DNA ligase, which is an enzyme that can connect 2 strands of DNA together by forming a bond between the phosphate group of one strand and the deoxyribose group of another, join together all the Okazaki fragments. This process is referred to as discontinuous synthesis. An enzyme called RNA polymerase separates the 2 DNA strands. The RNA polymerase must first combine with a region of the DNA strand called a promoter. The promoter region for a particular gene determines which DNA strand is the antisense strand. The antisense strand is the strand that is copied during transcription. The DNA strand that carries the genetic code is called the sense strand. The sense strand has the same sequence as the newly transcribed RNA, except with thymine in place of uracil. The process of transcription begins with an enzyme called RNA polymerase, which attaches to the template DNA strand and copies the DNA sequence towards the 5' end. The 5' ends of the free RNA nucleotides are added to the 3' end of the RNA molecule being synthesized. Polymerases are large enzymes that control the process of transcription, during which the information stored in a molecule of DNA is copied into a new molecule of messenger RNA. The RNA polymerase moves from the DNA promoter region towards the terminator. Along the way, it separates the strand while transcribing the nucleotides. The terminator is a sequence of nucleotides that, when transcribed, causes the RNA polymerase to detach from the DNA. When this happens, transcription stops and the RNA transcript is detached from the DNA. The transcript carries the code of the DNA and is referred to as messenger RNA. The mRNA has a 5' cap on the 5' end, and on the 3' end, there's a poly A tail. These caps make it easier for mRNA to leave the nucleus and protects the strand while also making it easier to connect later on. Overall, during replication, the new strands read in a 3' to 5' direction, which means they grow in a 5' to 3' direction. By the end of the process of DNA replication in the new daughter helixes of DNA, the parent strand is the one strand that is passed on from the original strand of DNA, and the new strands are the newly synthesized strands that are paired with the original strands.
Once mRNA is produced from the DNA template, the process of actually producing the protein at the ribosomes can begin. Each ribosome consists of a large subunit and a small subunit. The decoding of a strand of mRNA to produce a polypeptide occurs in the space between the two subunits. In this area, there are binding sites for mRNA and 3 sites for the binding of tRNA. The A site holds the tRNA carrying the next amino acid to be added to the polypeptide chain. The P site holds the tRNA carrying the growing polypeptide chain, and the E site from which tRNA that has lost its amino acid is discharged. tRNA moves continually through the 3 binding sites. The triplet bases of the mRNA codon pair with the complementary bases the triplet anticodon of the tRNA. The initiation phase. The start codon is on the 5' end of all mRNAs. Each codon attaches to a particular tRNA. The tRNA has a 5' and a 3' end. The 3' end of tRNA is free, which is where the amino acid is attached. One of the ends of tRNA contains an exposed anticodon, which is unique to each type of tRNA. This anticodon that pairs with a specific codon of mRNA. The active site of each enzyme allows a fit only between a specific amino acid and a specific tRNA. The actual attachment of the amino acid and tRNA requires energy that is supplied by ATP. The first step in initiation of translation is when an activated amino acid, methionine, attaches to a tRNA with the anticodon, which combines with an mRNA strand in a small ribosomal subunit. The small subunit moves down the mRNA until it contacts the start codon. This contact starts the translation process. Hydrogen bonds form between the initiator tRNA and the start codon. Next, the large ribosomal subunit combines with these parts to form the translation initiation complex. The translation is when an activated amino acid, methionine, attaches to a tRNA with the anticodon, which combines with an mRNA strand in a small ribosomal subunit. The small subunit moves down the mRNA until it contacts the start codon. This contact starts the translation process. Hydrogen bonds form between the initiator tRNA and the start codon. Next, the large ribosomal subunit combines with these parts to form the translation initiation complex. The elongation phase. Once initiation is complete, elongation occurs. This phase involves tRNAs bringing amino acids to the mRNA in the order specified by the codons of the mRNA. Proteins called elongation factors assist in the binding of the tRNAs to the exposed mRNA codons at the A site. The initiator tRNA moves to the P site. Phase. The termination phase begins when one of the 3 stop codons appears at the open A site. A protein called a release factor then fills the A site. It breaks down the bond linking the tRNA and the P site with the polypeptide chain. This frees the polypeptide, releasing it from the ribosome. The ribosome then separates from the mRNA and splits it into 2 subunits. The termination phase completes the process of translation. This process is repeated continuously, and the group of ribosomes used are called polysomes.